Gram-positive versus gram-negative bacteria, differences and importance. Bacteria are everywhere. They can be found in the air, water, soil, and even inside our bodies. But did you know that bacteria are classified into two main groups based on their cell wall structure? Today, we'll explore the fascinating world of gram-positive and gram-negative bacteria. But wait, what does gram-positive and gram-negative even mean? Let's break it down. What is gram staining? In 1884, a scientist named Hans Christian Gram developed a special staining technique called the gram stain. This method helps scientists identify bacteria by coloring their cell walls. Bacteria are stained with crystal violet, purple dye. Iodine is added to fix the stain. Alcohol is used to decolorize the cells. A second dye, safranin, red dye, is applied. Now, here's where the magic happens. Some bacteria retain the purple color. These are gram-positive. Others lose the purple stain and turn red or pink. These are gram-negative. Differences in cell wall structure. The reason for this color difference lies in their cell wall structure. Gram-positive bacteria have a thick peptidoglycan layer, 20-80 nanometers, biceps no outer membrane teichoic acids, which provide structural support retains the crystal violet stain. Appearing purple gram-negative bacteria have a thin peptidoglycan layer, 210 nanometers, an outer membrane containing lipopolysaccharides, LPS. Porins, which control molecule entry, does not retain crystal violet, instead takes safranin stain, appearing pink. Show a side-by-side comparison. This outer membrane in gram-negative bacteria makes them more resistant to antibiotics. Examples of gram-positive and gram-negative bacteria. Now, let's look at some examples. Gram-positive bacteria. Staphylococcus aureus can cause skin infections and food poisoning. Streptococcus pneumoniae causes pneumonia. Bacillus anthracis, the bacteria behind anthrax. Clostridium botulinum produces the botulinum toxin, leading to botulism. Gram-negative bacteria. Escherichia coli, E. coli, found in our intestines, but some strains cause food poisoning. Salmonella, a common cause of foodborne illness. Pseudomonas aeruginosa, resistant to antibiotics and found in hospital infections. Neisseria meningitidis, causes meningitis. Some bacteria are helpful, while others cause diseases, but both play essential roles in our ecosystem. Antibiotic resistance, why it matters. Did you know that gram-negative bacteria are often more difficult to treat with antibiotics? That's because their outer membrane acts like a protective shield, making it harder for drugs to penetrate. This is why antibiotic resistance is a major concern. Overusing antibiotics can lead to superbugs that don't respond to treatment. So, always take antibiotics only when prescribed by a doctor. Fun facts about bacteria. Before we wrap up, here are some fun facts about bacteria. The human body has more bacterial cells than human cells. You are basically a walking bacterial colony. Some bacteria can glow in the dark, like Vibrio fischeri. The world's largest bacterium, Thiomargarita namibiensis, is the size of a grain of sand. Some bacteria can survive extreme temperatures, from boiling hot springs to frozen glaciers. Amazing, right? Bacteria are tiny but powerful, playing roles in health, disease, and even the environment. Conclusion: why understanding bacteria is important. So, now you know the difference between gram-positive and gram-negative bacteria. Gram-positive equals thick peptidoglycan, no outer membrane, stains purple. Gram-negative equals thin peptidoglycan, outer membrane, stains pink. Bacteria may be small, but they have a big impact on our world. Understanding them helps in medicine, microbiology, and disease prevention. If you found this video helpful, don't forget to like, share, and subscribe for more amazing science content.